Have a good day, everybody. The Forest Linking Unit of the Forestry School of the Tecnológico de Costa Rica welcomes you to the webinar conference, Progress on Teak Genomics. Today we have three lecturers. One of them is Olman Murillo Gamboa. He is a professor and researcher at the School of Forestry Engineering of the Technological Institute of Costa Rica. He is a founder and director of the GENFORES International Cooperative of the Forest Genetic Improvement from 2002 to date. He is a Master of Science in Forest Genetic Improvement from North Carolina State University, and he also has a PhD in Forest Genetics from the University of Göttingen, Germany. Olman has more than 300 scientific publications, including books, scientific articles, manuals, and presentation and congresses, conferences, and international events. He is the specialist in forest genetic improvement, intensive forestry, and forestry evaluation and forest plantation evaluation. On an international level, Olman has several research and extension projects concluded since 1983 with the collaboration and participation of universities from Latin America, United States, and Germany. He has been a director or advisor of bachelor's, master's, and a PhD thesis. He is also advisor and consultant for FAO from the Fondo Nacional de Financiamiento Forestal, and also from forestry companies from Colombia, Nicaragua, Ecuador, Peru, Mexico, Guatemala, Brazil, and Tanzania. He also is a member of the Scientific Committee of International Journals and organizer and instructor in 58 training courses, national and international events. Our next lecturer is Mr. Andrew Callister. Mr. Andrew Callister is a Bachelor of Forest Science. He is a philosophy doctor from the University of Melbourne. He is the principal from the Treehouse Consulting and Treehouse Forest Research from the United States. He does a provision of R&D consulting services to plantation forestry businesses and research projects, specifically in the following fields: quantitative genetic analysis and tree improvement strategy, genomic prediction and relationship analysis, envirotyping and predictions of genotype environment interaction, seed production and supply of improved germplasms, carbon sequestration, accounting and gold standard certification, plantation silviculture research, and forest growth and yield modeling. And the last lecturer is Mr. Jose Pablo Jiménez Madrigal. He is a biologist and he's actually or currently a professor and researcher from the Escuela de Ciencias Exactas y Naturales from the Technological Institute of Costa Rica. He has a master's degree on forestry and a PhD on functional genomics from the University of North Carolina. So these are today's lecturers. So we will have some presentations, and what we are going to do is that they are going to present first, and at the end of the session, if you have some questions or if you want to discuss some topics, there will be an space so you can speak and ask to the lecturers all the questions or important topics that you want to ask. Bienvenidos, bueno, a los que hablan español, bienvenidos a este webinar organizado por GENFORES, ¿verdad? Sobre genómica forestal. Hoy vamos a tener tres presentadores. Uno de los presentadores es Olman Murillo, profesor de la Escuela de Ingeniería Forestal, fundador y miembro de GENFORES desde el 2002 para acá. Olman tiene vasta experiencia en todo lo que es la parte de silvicultura de plantaciones y la parte de genética forestal. Ha trabajado en gran cantidad de proyectos de investigación en este tema y así también ha sido profesor y director de tesis a nivel de pregrado, maestría y doctorado en estos temas. Después vamos a tener al señor Andrew Callister, que él trabaja para la empresa Treehouse Consulting, ¿verdad? Y él es, o se desarrolla su trabajo en la parte de análisis cuantitativo genético, toda la parte de ¿cómo se llama? De producción de semilla y distribución de germoplasma mejorado. También tiene experiencia en todo lo que es la parte de secuestro de carbono y la certificación del Gold Standard y en la parte de silvicultura de plantaciones. Y finalmente, el profesor José Pablo Jiménez, que es profesor investigador de la Escuela de Ciencias Exactas y Naturales. José Pablo es miembro de GENFORES y también es miembro del programa de doctorado Docinae. Y José Pablo tiene, bueno, una maestría y un doctorado de la Universidad de Carolina del Norte en la parte de genómica funcional. La dinámica de este evento va a ser la siguiente: los expositores van a tener un tiempo para dar sus presentaciones y después de las presentaciones se va a abrir una sesión y una parte de preguntas donde ustedes van a poder hacerle la pregunta a los expositores o discutir sobre algún tema de interés. Entonces, sean bienvenidos y le cedo la palabra a Olman.
Thank you, Mario. I will be okay. Let's move and welcome everybody. Many of you are my friends from lots of years. I'm very happy you are here. So what we wanted, the goal, the objective of this presentation is going to be because we wanted to share with you where we are exactly in this area of genomics, in teak genomics. We've been very active and busy on all these areas in general, tree improvement, but now getting into genomics as everybody's doing in this genetics area. So I'm gonna show you the information we have prepared for you. First of all, just let me put this into presentation mode. Now we go. Sorry. Okay. First of all, really quick about the research team, who we are now in GENFORES or GENFORES. This is the main group of researchers. We, the four, are part of the staff members of our School of Forestry Engineering. And well, pretty much who we are in the specialties in the area of genetics. So we have here Yoleni and Dawa.
No está compartiendo.
No está compartiendo.
No, Olman, todavía no podemos ver la pantalla.
Perdón. I'm sorry, let me see. I have to. I'm sorry, I have to. Wait. No. Aha. Now I have to do it like that. And what is now? How is now?
Ahora sí lo podemos ver, Olman, sí.
Okay, it's because I did not. Yes. Okay, now we are here. Okay, I'm sorry. Okay, so let me see if I can take this down because okay. And how can I take down this information? Because well, let's go. So as I was talking here, this is our group, our main group, and we have 4 full-time members of our faculty of forestry in the cooperative, assisting the cooperative research, all the duties we have to do with the cooperative. So Yorleni Badilla, she's mostly with the clonal forestry development and quantitative genetics. And then Dawa, she's working on genetic resistance. And Jose Pablo, he's basically in charge of all the quantitative and genomics. And then we have another associated researchers from other institutions in the country, like especially from the National University of Costa Rica, which is another public university. And there we have Anna Hein, who is a specialist in biotech and pollen management. Actually, he did all her PhD on that area, developing all needs we have about pollen in general, storing pollen, and etc. Then William Hernandez, another good guy, a new member of our research co-op, is mostly in quantitative genetics. And Diego Jiménez from CATIE is now a member of our co-op, institutional member. And then Emmanuel Araya from the National Institute of Biotech in Costa Rica, related to the public universities. This Emmanuel is mostly on genetics and, molecular genetics and biotech. Now the members, the active members, this is something in the history of our co-op very quickly. We've had some, like used to happen in the companies. Now the very active members are these 7 guys. We used to have cooperatives, Costa Rican cooperatives. As you may know, Costa Rica is a very socialized country. So we had strong cooperatives or NGOs working in forestry. They are no longer active members. They used to be. Same thing for a very nice Brazilian company we used to have here. They are not any longer active. So that's why I'm not posting them. So they are company members, so like traditional, in the traditional way of this type of cooperatives. We as a research institutions, we are 3 Costa Ricans, all of us, well, the first 2 are public universities, and CATIE, well-known CATIE, the international institution in Costa Rica. Associated companies getting into the forest, into GENFORES, we have a process to admitting new companies. We're always open to that. So they have to follow like a 2 years process just to be, we want to know if they really want to get into tree improvement, real tree improvement, to put resources and efforts in that area, because we've had a lot of stories about this. Not everybody is really pushing in a very disciplinized way to get into real genetics. It takes time. And now recently, very recently, from the last years, and thanks to Andrew Callister, Dr. Andrew Callister, we are now working with Tanzania, this Kilombero Valley Teak company, very well known for many people in Africa. This is the largest teak company in Africa. We started a new collaboration with them. We are working together in clonal, trying to move the guys into clonal forestry, for teak clonal forestry. And Andrew this is one of our lectures today. He's going to be presenting at the end of the presentation his participation, because we invited him to help us with the data analysis. Andrew is an expert in these areas of genomics and data analysis. So he's going to be as well as myself in this presentation in the last part of the genomics analysis. Now, the breathing, what is going on with breathing? I want to stress this is very important. We have to relate or base whatever we do in genomics to breathing programs, real breathing work, real tree improvement programs. This is what I just wanted to stress. This is a general view of what we have, the number of accessions we have, almost 400 accessions. This is probably the largest breeding program in teak in this moment in the world. I guess, I'm not sure if there are any other group like as big as this. So you see the companies, the number of accessions per company. We share tests, genetics tests, and so on, and we try to exchange also genetic material. Now we will be moving into exchanging pollen, not trees, but pollen. It's going to be a lot easier. And then the number of genetics tests, every year we have new tests, some of them coming in and coming out as normal. We've been testing for 18 years, and we have a lot of history on this genetic testing, teak as well. More than 20, 30, some years we have 30 active genetic tests everywhere. So this is a very active area. Now I have here some of the basic easy ways to start doing breeding. We have here our good friend Faustino Takizawa from Floresteca, the largest company, private company now in the world, the largest teak company in the world in Brazil, in 2 states of Brazil. And we have been talking about getting into GENFORES as well. Because of the COVID, we stopped the relationships and the discussion, but we will probably continue talking together again. So this is our +3 selections, the initial, as you may know. The idea is to relate those super trees to the genomics. This is going to be happening today. We will be explaining you how we are trying to get the information inside the genetics from those super trees. This is a nice view from one of the best companies, to me, one of the best companies in the country, and of course in GENFORES. This is Pan American Woods. They established their operational clonal forestry in year 2004. They are all actually now getting into they're finishing the first rotation of a completely clonal plantations. They're getting into something like 18 years old, something like that. And Floresteca and Pan American Woods are probably the first 2 big companies who are doing clonal forestry at operational scale. Probably the first 2 at large scale, planting clones. We are very active in getting into the next breeding generations, of course. So we have been doing a lot of research on this, trying to promote flowering, to do easier and faster. These are not teaks, please. I'm just showing you the research. We're ongoing research on teak and melina, and these are melina, which are very easy to promote flowering. We are doing the same thing with teak. We got a lot of new ideas from Brazil, from the eucalyptus. So we train our people in Brazil in these areas. Was part of a PhD from Jorleni. She got recently her PhD on that. And we are now adapting these technologies to teak. And we are very close now to get that. Pollination techniques and several issues about these pollination techniques, how to do it operationally. Because pollination has been a long time, quite well known. The Asian guys, the Asian scientists already published a lot about this, but we're now moving into operational scale, which is quite different. Now, pollen, as I mentioned, we had to learn a lot about how to do the pollen handling, storage, and analysis, and so on. If you don't do this, you can't go further in next breeding generations. So we are very active now in this, getting into the second breeding generation for teak. Actually, Pan American Woods is probably the first company at they are already in the second breeding generation, testing their new materials. And genetic resistance has been a big issue, getting more and more important. This is not teak, this is melina, but in teak we have pretty much the same type of diseases. We learned how to do genetic resistant tests. We got a lot of collaboration from Federal University of Viçosa in Brazil. And then we really got it. The Brazilians had a huge experience on handling, overcoming diseases in eucalyptus and pines. And so we learned a lot from their teams. And we had a very strong collaboration with Brazil in this area as well. This is another area we've been very active as well, these wood properties. Led by Dr. Moya from our school, from our staff members, research members in our School of Forestry. And he has been very active with us, helping us with all these topics around color, hardwood color, and other specific gravity, other wood properties. This is a very important area for us in breeding for wood properties in general. Color is going to be a very important issue. It is already in marketing teak wood. And then we do have our own molecular genetics lab. We established this about 20 years ago, and we are been pushing and trying to clean up, let's say, all these issues of gene markers and microsatellites. Very busy. Sometimes they're not so stable. We had to move on that, how to improve the gene markers lectures. Sorry, the information they provide, not too stable, not too happy. We still have them. We are doing a refinement of these microsatellites information they provide. But of course, now genomics is getting in, and then it's going to be probably part of the history. We are probably moving our lab to genomics now that we have a lot of information on genomics. This is the normal history of all this area of gene markers, population genetics, molecular genetics are getting really moving very fast. Okay, so that's pretty much my presentation. I will be talking at the end just to telling you what is next, what is going to be in the next couple of years. So I will introduce Jose Pablo, and he is going to talk to you how to do this. Okay.
Hi, everyone. All right. Start with this. So as Olman mentioned, I will be talking about DNA sequencing and marker discovery. Now, I was asked to provide a basic introduction to genome sequencing, as well as some of the related work that has been done in general, and especially from our co-op. Now, this might be too basic for some of you. If it is the case, I do apologize. I can go into more detail on the results and more depth into the techniques at the end of the seminar, the webinar, if you want to. If this is all new to you, feel free to ask any questions. I'll go back and go over it. But then again, it's more like a basic primer on DNA sequencing, so you understand how we get this data, and then Andrew will go into more detail on how we apply that data, how we can use it meaningfully. All right. Okay. So the first thing that we need to talk about is what is a genome, right? Because sometimes we use genome, we use genomics and so on, and it sounds like nice, but we don't really know what it is, right? So we do understand, right, that genes are the hereditary units that are transferred from parents to progeny. We know that since Mendel, right? Those genes lay the information or have the information to build all the enzymes and all the proteins needed for the cell to work. Right. And that information is encoded in a molecule, a chemical molecule called DNA, right? So we call the genome the entire set of instructions written in that DNA that we can find in a cell. We can find it either in the nucleus, or you can find it in the organelles like chloroplast or mitochondria. Right? Now, traditionally, when we talk about the genome, or the traditional view of the genome is something like this depicted here, right? That karyotype, like the chromosomes, things that we could do, we can actually stain them and look at the microscope, right? But nowadays, when we talk about a genome, we're talking more about the information content, right? The actual sequence that we can read. And it turns out to be like text files, right? That we can see the A, G, T, G, right? All the letters that conform that. So quite literally, it's become like a book that we can read, and it gives us some understanding of how things are built or how things are done, and how things are done by the cell. So since it's like text files, we do a lot of bioinformatics with it. We do a lot of computational analysis to get meaning out of that information. And just to put it in perspective, genomic data has now become part of the group that they call big data, right? So we're right there with astronomical and social media due to the magnitude of data that we have. We're talking about, for instance, the human genome. That will be a book, or that will be a file with 3 billion characters, right? That's a huge file that we need to manage, and you probably won't be working with just one individual, right? You're looking at difference, you're looking at data of several individuals, so it becomes more complex. Now, how do we get that data? How do we get that sequence? How do we know exactly what's the order, what's saying in that molecule, that DNA molecule? For that matter, we use sequencing technologies, right? And the first generation of sequencing technology is what's called the Sanger sequencing, where we use an analog of a nucleotide, and then by termination, we can determine the sequence of the molecule. Later on, we atomize that, and what we do is we use nucleotides that were labeled with a color, so we can read that with a laser, electrophoresis event, and we can determine each of the bases on the DNA molecule, right? We can sequence and we can get an idea. This is how the human genome was sequenced, right? We use a bunch of this equipment to sequence small pieces of the genome and get that information out. It's still considered a really good technique. It's just that it has some limitations in terms of the throughput, or how much information you can get out of equipment, how much information you get out of any given reaction at any point. So enter the second generation of sequencing technologies, what we call next generation sequencing, next gen, NGS, or it's also called high throughput sequencing technologies. The improvement here is that we do the reactions in parallel. So instead of having one machine sequencing one piece of DNA, we have millions of pieces of DNA attached to a small cell, and we can go ahead and read in each one of those. Right now, this type of second generation or next gen sequences is dominated by Illumina. That'll be the company that provides the services or builds the chemistry and the sequencer, the equipment that they use. You probably will hear from those different vendors that provide the Illumina sequencing or PacBio sequencing or whatever. But this is just the technology. It's right now the cheapest technology to sequence. Cost has dropped significantly, so you can do it, but it has the limitation on the size of the fragment that we can read, right? So we can sequence small fragments, but we can sequence millions of small fragments at the same time, right? So you get a lot of information out of it. And then there's a third generation of DNA sequencing technologies that will be the single molecule sequencing. And that's what PacBio or Oxford Nanopore equipments do. The advantage of this is we're still sequencing a lot of molecules in parallel, but we can get longer pieces. So if you think of the genome, as I was saying, like a book of instruction, if you're doing Illumina, you're sequencing one word or one sentence at a time. If you're doing PacBio, you're doing pages or chapters at a time that you're sequencing. So if you have to put that manual together again, it would be much easier if you have the bigger pieces, right? But again, it's just a type of technologies. It has different prices and it has different applications depending on what you want to do with them. But just a basic primer, you can go ahead and read a little bit more into it, any of those if you're interested in. Now, what has been done in teak? Well, before we only have the available data it was bits and pieces there, partial genes that were sequenced mostly for phylogenetics, a lot of work done with molecular markers, SSRs or microsatellites, right? So there's a lot of sequences of microsatellites available in the databases, organelles, genomes. So for instance, the chloroplast genome is sequenced, the mitochondrial genome from teak is sequenced. But again, it's small information that we cannot use as much for breeding or for further genomic applications. It is until 2018 with the Ramazani Yosoda publication that they get the first draft of the first draft assembly of the teak genome. Right? So this is a fairly fragmented assembly, but it's a complete assembly of the teak genome. It has valuable information that we can use. And in a rapid succession, like shortly after in 2019, Sao and collaborators, they published a chromosomal scale assembly of the teak genome. So this is a far more complete sequence, less fragmented. I mean, it's at the level of pseudo molecules or chromosomes. So this will be considered right now like the reference This is the sequence that we use to as a reference for any other studies moving forward. And shortly after that, 2021, the teak genome was published by the Springer Nature. This is a book that compiles most of the most current research done in teak genomics, including the teak genome as it is, but also functional genomics, a little bit of history, and what has been done in terms of molecular advancement in teak research. And this trend continues. I mean, there's a lot of new information out there, and every day we're generating more genomic data that we can use for tree improvement or for basic conservation that we can use. This is a recent screenshot that I took. I think that it was a couple of days back. Just to give you an idea, this is from the NCBI Gene Bank, one of the most known and available freely available databases for genomic and genetic information. And if you can see there's more than 3,000 nucleotide sequences available there for teak. As I said, many of those are partial genes or microsatellite sequences, but also we have hundreds. Right now we're in the 725, hundreds of genomic sequences. So this is whole genomes that have been sequenced and the information is put available. This is reduced representation that we're going to talk about a little bit ahead in the presentation. Well, there's a lot of information there right now, and I bet that it's going to continue to grow, and more information is going to be available because sequencing and genomics is dropping in price. And my advisor is in the audience. My former advisor is in the audience. He's probably going to scold me for using this. He used to say this is one of the most used slides for people working in genomics, but it just comes to show how drastically the prices has dropped, right, since the beginning of this. So just to put it in perspective, what you have there is 2001. That's roughly when the Human Genome Project ended. Officially, it ended on 2003, if I recall correctly. Right? But it started in the 90s, right? And since its inception to 2021, which is the most recent information, you can see it has a drop of on the price of or the cost of sequencing a genome has been quite significant. Right? So right now, well, for instance, when the human genome ended, it costed roughly $2.7 billion to sequence. Nowadays, you can get a whole human genome for $1,000 or less. In comparison, if we were talking about teak, you can get a teak genome low coverage ranging from $350 to $1,000, depending on if you're going to do all Illumina short reads or you're going to do PacBio long reads. Also, depending on the service provider, right? The service providers are throughout the world. The prices change. Sometimes they have discounts and so on. So you might get this a little bit cheaper. It might be a little bit more. But the thing is roughly about $350 to $1,000 to get a teak genome, like a whole teak genome data information. Now, you have to admit that that's quite a drop in price compared to what it used to be, but it's still higher than you could use commercially, right? If you want to implement, if you want to be sequencing thousands of individuals, hundreds or thousands of individuals, it's still a steep price to pay $300 per sample, right? So what we can do to reduce this cost is what we call reduced representation sequencing. So we're not going to sequence the whole genome, but we're just going to take bits and pieces throughout. So we have homogeneous and sort of representative part of the genome being sequenced. And there are several techniques that you can do for that. One of those will be what we call RAT-seq, or restriction site amplification sequencing. And the other one is called GBS, or genotyping by sequencing. You might hear both terms used interchangeably. I think that the main difference, I mean, there's some methodological difference there, but I think that the main difference right now is that GBS has a patent on it. So if you use this service commercial, you might have to pay a little bit more for GBS than if you do your in-house RAT-seq experiments. But how this works is basically you take the genome and you cut pieces with restriction enzymes, and then you just sequence a little bit from that cut site on the genome, right? And those enzyme cuts are supposed to be uniformly distributed throughout the genome, so you get a good representation. And you can test with different enzymes just to get an idea of how much of the genome you're actually capturing, right? If you want to capture more or less of the genome without having to sequence the whole thing. And then again, I mean, there's plenty of examples of this type of data populating nowadays the databases. Just recently, I checked, let's see, in February of 2023, people from the UniMed uploaded to the GenBank database on teak SNPs generated by GBS, and that was 211, I think, individuals. Before that, 2021, the Wageningen University also provide a rat-seq experiment with 37 biosamples, so on. So there's more and more data that is being constructed. And then the idea with this is that we will go ahead and cut the genome, sequence just small pieces. But the thing is that we know those pieces, we can align them to the reference. That's what we call, let's see here, okay, get a winner. So you just go ahead and cut it, add an adapter so we can sequence. We knew that adapter will also usually include a barcode sequence so we can identify every individual that we put into the sample, and that we can pull samples. That makes it more efficient as well and reduces the cost. And then we sequence those and we can align them back to our reference, in this case, probably Sao's reference. And then we can compare between individuals. Let's see, we have it here. This is from Stacks. That's a software that we can use for this. So what you can see is that we can go ahead and sequence all those pieces, right? We can make stacks or group them depending on the region of the genome that we were sequenced. And then we can go ahead and compare those, right, per individual and identify those differences that are between one individual or the other, right? And those differences in this case is what we call SNPs, right? SNP markers. The SNPs are really abundant throughout the genome. Those SNPs is a mutation on a single nucleotide. So part of the sequence that you're reading, let's imagine that you just change one letter, and that letter can have a significant change or not on the meaning of the gene or the transcription that you get, or it might not. It might be in a neutral part of the genome. It might be part of the coding regions, the part that actually is translated into proteins. But then again, I think that the most important part is that they're really abundant. That throughout the genome of any species, they're really abundant. We can use that to associate with traits, and that's something that you're going to see later on this webinar. And just for fun, if someone has doubts whether one letter can actually change something, like if you have one SNP, could it actually change the expression of a gene? Let's just run this quick experiment. This is the word bar. It's very simple. Bar is a place where you go and you can have drinks, right? If you change one letter from B to a V, and if you've been drinking, you'll be forgiven to change or mispronounce it or misspell it, right? But technically, this thing, I mean, you still understand the meaning, right? Like if I say bar or var, I mean, it sounds funny, but you still understand the basic gist or the idea, right? But if I change again to one letter and I change it to car, that's a completely different word, right? I changed completely the meaning of that. You can drink in a bar, but you should never drink in your car, right? It's the same with genes. It's the same with the genome. You change one letter, and that might have no consequences. That's what we call a neutral mutation. Or you might go ahead and change the meaning of that. It might change the protein that you produce, and that mutation might be beneficial, might be detrimental, right? It might be associated with a phenotype that we're interested in using, right? So SNPs are important. SNPs are really abundant, and we can use them to predict or use it to work on tree improvement, right? So what has GENFORES done so far in this respect? Well, we've worked with genome sequencing. We sequenced 2 of our teak elite genotypes, right? Generated that completely from short read Illumina reads with 180 coverage. That's probably overkill, but it's a really good coverage. Just basic information there, FastQ and BBDoc for quality control of the reads. And then this was on the de novo assembly using spades and error correction with RACTech. And for error correction, we use the SAO reference just to help with misassemblies and so on. And we ended up with 2 assemblies that are partly fragmented, but they're complete. I mean, there's a good representation of the human gene, of the teak genome in this, and therefore operational purposes, they were pretty good as future references that we can do. However, probably the most interesting part and relates with what we're going to hear later on is the experiment that we did in RATseq, right? Reduced representation. As I mentioned before, ideally you want to sequence the whole genome of all your individuals, but that's an expensive proposition. So you have to reduce costs, and the way to doing that is doing reduced representation. So in this case, for us, the price per sample roughly goes by $50 if we're doing 96 samples. However, those prices are probably going to drop because this is a scale economy. The more samples I do, the more efficient that it is. So that the less the cost per sample that you can get. And also keeping in account that at least for GENFORES, we don't have a sequencer in-house. So we outsource a service and we have to pay the shipping and so on. So that kind of plays a little bit on the prices. But still, $50 per sample is more doable than $350 per sample, right? And if you look at the amount of data that we get from this, the amount of SNPs that we were able to identify, it comes to less than a penny per SNP, right? So that's a great, I mean, that's a bargain if you're looking at that, how much data you're getting, how much it's costing you, each of those marker data there. Right. So for this experiment, what we used was 33 genotypes from the lead collection. We also did some RATseq with control crosses. I'm not presenting those results here. Let's just go ahead and concentrate on the lead collection. Right. Library preparation was used using the PSC1 and MSP1 enzymes for the copy. And again, this was sequence with an Illumina equipment short read sequence pairing 150 base pairs. This work was done by collaborators in North Carolina State University. Dr. Ross Watt, that is in the audience, he helped us a lot with this, with the library preparation. However, we have the tools to do it in-house now. That quality control for the reads is again FastQC and BBDocs. We align using BWA and SAM tools, and we use the SAO reference, the SAO assembly as a reference. You can do marker discovery de novo without a reference, but it's going to be more robust. You're going to get better data if you can associate it or you can align that to a reference. So that's the importance that someone already created one, right? We started this project. I show you we create our own references as well, our own sequence genomes. But then again, SAO is so good that it would be a shame to pass on it. But and then we do the variant calling for this one. We use PCF tools and then filtering, right, with certain parameters that you have to work. And then we end up with 10,816 SNPs. This is what we call in the range of a low to medium density marker panel. I mean, we're not quite there with the 50K that people in eucalyptus use, but it's fairly good. I mean, you get a good representation. It's 10,000 markers that are distributed throughout the teak genome. And just full disclosure, the actual number of variants that you can detect in teak in our sample is way larger. This is roughly like a 50% of the less than 50% of the markers that we actually identify. What happens is a lot of those markers are dropped out of the panel because of missing data, and that's something to do with just artifacts of the technique, sequencing errors, and so on. There's another part that is dropped because there are different variants. We're only interested in SNPs as it is, like single nucleotide polymorphisms. Sometimes you have variants that are more than one nucleotide. And we have indels, right? Insertion, deletion, mutation that you have there. We usually clean those up. You could use them eventually, but for the models that we use nowadays, it's way easier if we just work with SNPs. And we want to work with SNPs that we have information for all the individuals. And that's another thing that is the nature of working with a breeding population that has a fairly limited genetic diversity. We end up with a lot of individuals that have a significant amount of what we call private alleles, right? And you're going to see in a minute when Andy starts his presentation that there are distinct groups within this data sample, within these genotypes, right? There's small populations that are separated from others, and there's some alleles that are only present in one individual or the other, right? For the general purpose of modeling, that doesn't work that well. Having data for just one individual and blanks for the other. So we drop a lot of that information. However, if we expand our sample size, the more individuals will include in our database for future rat-seq experiments, the more markers we're going to get data for, and the more we can increase on this panel. But then again, 10,000 is a good number. It's on the low side, but it's still usable for modeling. Right? And that brings us to what are the potential application of these markers. You can use this to estimate diversity. That's a very important thing for anyone working in breeding, is knowing the diversity, the genetic diversity of your populations, your breeding populations. Traditionally, you can do that by estimating heterozygosity and other metrics. You can still do this with the genomic data. It's just that you can go even deeper and you can do nucleotide diversity. So you can calculate the pi, like what is the nucleotide diversity in my population? You can calculate divergence, right? How much each population, or how much each cultivar or each clone differs one from the other. We can use this for provenance or clone identification. So if you want to make sure the clonal purity of your banks, you can run this sort of experiments and try to find the relationships between individuals and make sure that your clonal bank is actually what is supposed to be. Right? It's the idea is there. You can do genome-wide association studies to try to connect this marker with a phenotype in particular that is interest, right? And by doing that, you can also then enable marker assist selection, and eventually what we're hoping to is get to genomic selection. And any of these techniques that we use is not only going to help the improvement by improving the quality and the precision of our estimations, but it also could reduce the breeding cycles, right? By enabling early selection. And Andrew's going to go into much detail on these topics. Thank you.
That's your cue.
Andrew.
Yes, we are going to have Andrew now, Dr. Andrew Callister. We will be moving into a more, well, strong analysis data. So we will have now Australian accent. I hope all of you can follow him. So thank you, Andrew. Go ahead.
Yeah, thanks, guys. Less than a penny a marker, Jose Pablo said. I think that's a great marketing line. I can see you getting a lot of work coming your way. So we, of course, want to use these genomic markers in a predictive capacity. We want to be able to use them to conduct an early clone selection or to screen candidate clones before they've been trialed. And this is the focus of my presentation here. The goals of this work that I'm going to present were firstly to reveal relationships amongst the clones that were sampled, and then to use two different methods for genomic prediction. The first being a Bayesian regression method, or called Bayes alphabet model, and the second being an association study or genome-wide association study, and then evaluate both methods for the potential commercial applications. So the data came from a single clone trial in Costa Rica. There were 33 clones that were genotyped and 36 clones that we had clone genetic values from phenotype values. And 26 clones were in common to the genotyped and the phenotype data sets. So let me pause for a second and just say that 26 clones is not a lot. It's a really good start. Ideally, with a study like this, we want 10 or 100 times that number. So take these results in that context. This is a good start, and we've got some promising results here. So the experimental data, the phenotypes that were collected on the 36 clones were tree quality, which is a combination of form attributes that GENFORES puts together, diameter, breast height, and volume, wood-specific gravity, heartwood percent, and then 3 color attributes, that come from the C-Lab color system for the heartwood color. The genomic data came in really good shape already when I received it. I only removed 4 markers that had missing values at more than 10 of the clones. And I used a imputation process to impute the 2% of data that were missing before analyzing it. And so the first result I'll present is the relationships amongst the clones, and for that I used an R package ASR genomics. The figure here is a heat map of relationships. The yellow color means there's no significant relationship amongst the 36 clones here. And you can see little squares of orange. And those squares indicate that there are significant relationships, and the values of the relationships in this matrix suggest that they are actually full subfamilies. So the genomic data already revealed something interesting. Amongst these 36 clones that we didn't know previously had any genetic relationship, there's actually 7 small full subfamilies in there, each full subfamily containing 2 or 3 clones. And 5 of these full subfamilies are also present in the data set that we use for genomic prediction. So we're going to have to deal with that when we come to the genome-wide association. So the first of the genomic prediction methods that I used was a type of Bayes regression model. And there's 5 variants of that here: Bayes A, B, C, lasso, and ridge regression. They're all similar in that they're all trained on the entire marker data set using the clone values. So it's not looking at individual marker effects per se. It's taking the whole SNP marker data set and building a training or prediction model. And the difference between the 5 different models, the different assumptions that they make about the distribution of marker effects. So they're relatively minor differences, but what we do is in common practice is we fit them all and see which one is performing best. And then the predictive ability, which is a result that I'm about to present, the predictive ability was determined by a leave one out cross validation. And what that means is that I take each clone one by one and mask its clone value, and then predict its clone value using its genotype, using its genomic data, and the Bayes model, which is informed by all the remaining 25 clones in the data set. So it's a way of taking each clone one by one, masking it and predicting it, the next clone masking it and predicting it, next clone masking it and predicting it. And then we see how well correlated those predicted values are with the actual clone values from the trial. And the result is summarized in this table. The red values are for Bayes ridge regression method, which for 5 of the traits provided equally best or superior predictions compared to the other types. So from now on, we'll just look at Bayes ridge regression rather than the other 4 Bayes model types. And we got pretty good predictive ability for form quality, for volume, diameter, specific gravity, and heartwood percent. And we got really rubbish predictive ability for the color traits. Negative values there are a little bit curious, and I think it comes down to just a very small sample size of only 26. So we're going to just basically ignore those. And then the right-hand column of joint selection is where I said if I was going to select the top 5 clones based on clone value, how many of those top 5 clones would also be selected as the top 5 from the Bayes genomic prediction. And so for quality and growth traits, 3 of the clones were in common, and for specific gravity and heartwood, 2 of the clones would be in common. So that's it's a pretty rough metric, but it's an idea that we're generally going to select the right clones by genomic selection using this method. Well, let's have a look in detail at diameter to begin with. So this is a plot where each of the points is a different clone. And we've got the predicted values by the Bayesian y-axis and the actual clone value from the field measurements on the x-axis. And you can see those 3 clones that would be selected in common up on the top right-hand corner. Now, unfortunately for diameter, the distribution of values by Bayesian prediction is a bit skewed. It's kind of concentrated in the low, the bottom half of the graph. So it's not ideal, but overall we're getting some kind of relationship there, considering it's a fairly small data set. By comparison, for specific gravity, the data are better distributed, and we still have that general correlation from the bottom left to the top right of the graph. And I've highlighted the 2 clones that would be selected by both genomic prediction and also by clone value. So the second prediction method that I used is genome-wide association study. And this is a way of looking at markers individually and their association with the trait of interest. The model accounted for, it had to account for population structure here, because what we want to do is identify markers that are significantly associated with the trait directly, rather than being indirectly associated via family effects. And I'm not going to get into the weeds on how I did that. But for people who are familiar with this field, you know, that's an important point to make. So firstly, how to interpret the results. I'm going to give the results in this table, and I want you to know how to read it. So for volume, for example, I discovered 11 significant markers. So these were SNP markers that were statistically significantly associated with the trait of volume, clone values. Unfortunately, 3 of those were completely collinear. And what's that mean? That means that 3 of them had completely the same results as another marker. So they were providing no new information. So in brackets 8, we have 8 markers there that are contributing some novel information. The false discovery rate is a theoretical value of how likely is it that a given marker that we think is significant actually is not significant, and that's calculated by the statistical significance of the markers and how many we have. And then in the right-hand column, I've got a value which is the proportion of the clone value for the top 4 clones that is captured by the significant markers. So for this case, in this case, 97% for volume means that we've got a good alignment in the top 4 clones between those that would be selected by the marker association method and those that would be selected based on their clone value. So here's the complete table. And what we can see is that by genome-wide association or by marker association, the traits that are looking promising are volume, that we just went through those numbers. Also specific gravity, which has 8 informative markers with a 57% false discovery rate and capturing 89% of the clone value in the top 4 markers. And color A, which has 5 informative markers, 47% false discovery rate, and 96% of the top 4 markers, the top 4 clones being captured by the marker information. So I'm going to drill in more detail to the trait of volume and give that as an example. So the first thing, every talk on genomics has to have a Manhattan plot. The Manhattan plot is a plot of statistical significance of each particular marker. So each of the dots here is an individual marker, and any of the dots that occur above the dashed line are statistically significant. So we can see not sure if the marker, if my cursor is coming through, but on chromosome 7, there's a point which is, so that's a marker on chromosome 7 that's particularly significant. And then also on chromosomes 10, 15, 16, they're also highly significant markers. And then some that are less significant are scattered across the chromosomes as well. The next result are box plots, and each of the individual panels here is representing an individual marker. The 0 on the X scale is the homozygous dominant state, and the 1 is a heterozygous state. And on the Y-axis, we've got the effect on predicted clone value of that marker. So for the first one, we can see that the center of the box plot is roughly 0.15 units of clone value higher for the heterozygous state than for the homozygous state. And that's an effect, that's our estimation of what the effect is of a particular marker on the trait of volume. So what we can do then is aggregate all of these effects and work out what the a genomic selection index might be for a particular clone, depending on what state their markers are at each of these markers, whether they're homozygous or whether they're heterozygous. And that kind of aggregation is presented for volume and for color A on this slide. So for volume, we can see a whole lot of clones are aligned at the value of 0 for genomic selection index. And they're just forming a line across the bottom of the graph for volume. And so these are all clones that were all homozygous for these significant markers and registered no genomic value for the trait of volume based on our marker association. And then the other clones are kind of scattered, including the 4 or 5 that are high up on the plot, which have more heterozygous markers and therefore higher values. So it's not a very fortunate distribution of values. And again, we'll come back to the fact that we've only got 26 clones here. And if we were to add another few hundred to the mix and redo the experiment, we'll certainly see the distributions of values. And again, just showing the potential of this approach, but not necessarily that this is the endpoint yet. So in conclusion, as I said, I'm going to repeat this again. The results are promising, but preliminary due to the small sample size. And, we've got some good results there for especially volume and specific gravity, which are traits that were both were predicted by both methods. The trait of color A from the C-Lab instrument was very well predicted by genome-wide association, by marker association, but had no predictive ability at all by the Bayesian method, which looked at all of the markers in the broad population. So that may potentially suggest that this trait is controlled by a small handful of genes instead of many genes across the nearly 11,000 markers that Jose Pablo provided. Future work has to be to increase the number of genotyped clones and also to increase the environment so that the phenotype information and the clone values that we get are more robust across environments. I'm keen to combine the Bayesian predictive and GWAS approaches into a unified model. Some other researchers have published some work providing a more unified approach instead of separate ones. And Jose Pablo is going to go on to annotate these significant markers that I found to see whether they are associated with genes that have got known function on the genome. And my final point is that once we do all that work and we strengthen it with a large number of clones, then GENFORES is in a good position to evaluate untested clones. So potential members that want to join the cooperative can put forward clones that they want to contribute to the cooperative, and they can be screened straight away using the genomic prediction instead of having to wait the 8 years or so for them to go into test and then be measured. And we can also shorten the time to clone selection. And this is especially important for wood quality attributes that take some time to be measured in the field. With that, I'll pass the control to Olman.
Yes, thank you, Andrew. I will be then at the end of this conference. Let me go. I'm sorry, I need to, I'm sorry, let me take this. I need to go there. I go to the Zoom again, and then we'll share my last part of this presentation. And let me check this moment. Now. So now.
Olman. Olman, está silenciado.
Yes, because I was trying to move this. Now I got it. I'm okay, right? Okay, what is next? And let me just try to finish. I think thanks to Andrew and Jose Pablo, of course. So I just wanted to say that I forgot to mention that this is not easy. This is not expensive to get into all these areas of advanced research like genomics, advanced breeding generations. Beautiful information is coming in from this analysis of data. For example, Andrew just mentioned that in our breeding populations, we were thinking that there was a possibility, but now we are certain that some of the trees that we call plus trees in one single company, for example, many of them are going to be related, very or strongly related, coming from an excellent and super mother somewhere in the past. Because we always do the tree selections, we do the plus tree selections, always in companies, in plantations, in commercial plantations, but you don't know the origins of those trees, where they come from, where did they come from. So what is going to happen with all these genomics is that now we have a lot more information to understand what exactly we have in our collections of what we call plus trees or tree improvement programs in each company. Now, the approach of a cooperative is extremely important to solve such situations. Not always bad, but in terms of relationships and what really the genetic diversity you have for the long-term breeding work, you have to know exactly what you have. And this is what I really think, this is where the cooperative approach is going to help a lot. You may have one company may think, well, I have 50 plus trees in my program. Maybe they are not really 50 itself different trees. You have many of them are half sieves, full sieves, as Andrew found, some of them are really full SNPs. It means they come from the same mother and father. So all these issues are coming, and we will be getting into what Andrew was mentioning. We need to sequence more genotypes. We need to have a broader, much more genotypes to run again all this data analysis, just to see if we can increase the predictability or the genetic correlations, let's say in general, how to predict if a tree is really going to be good in the future. For example, for the, Andrew was mentioning very well, the especially wood traits, which you can't know at the beginning in a tree, like a tree with 3-year-olds in your test, you can't tell at that time if this tree is going to be having an excellent wood, hard-to-wood color, or some other traits. For example, of course, commercial volume per tree probably is the #1 trait you want to know. If with some trees from a very early selection, you can tell which of them are going to be the winners, which of them are going to be the real superior trees and will be producing large benefits for the companies. So we have to continue working on this. For us, and maybe we didn't mention where the money came from. I think I was a mistake from myself. I want to just to remember, we got the funds from our university, our public university, and of course, as always, from the co-op members. It has been very helpful, very good. In this particular study of genomics, we got a very nice collaboration and help a lot from the guys from North Carolina State University, especially Dr. Ross Witten, and the genome sequencing lab, or I don't remember exactly the name, official name you have. So you can do all these things only if you have a very strong cooperation in between the companies and the academy. It is not possible to do this. I mean, if you are working alone. Now, so we will be probably, I'm sorry. We already have a ongoing research on this same area. We are now focusing on how to genotype more, to sequence more trees, more genotypes in our trials. We will try to get around 100 and see if the numbers will be going higher, the predictions. And again, the SNPs will be done again, and then we will go through, Andrew was doing at the end, associating the SNPs to the traits, the important functional traits we're interested in. That is pretty much what, that is pretty much what we wanted to share with you, and just to mention where we are, what is next, what is coming in the very few years from now. I think we will be moving very fast in this area. We now have the trained people, the skills, and the team. The only way to approach all these new developments in genetics. So I thank all of you for taking your time to hear us, to understand our different Englishes. You notice the differences in English, of course. And yeah, I apologize if my English is not the best, like Jose Pablo and Andrew. They are really bilingual, man. They really speak in a very good English. Anyway, the important thing is that we are sharing our research results and where we are. Well, more than research, we are really working tight with the companies. So this is the way we see these approaches or these developments. So if you have any questions, you can now talk with us directly. Orally, you can raise your questions, and we will go one by one and see what your information, your doubts, or whatever you want to ask. So thanks again, and I start sharing with you the questions.
Maybe I can jump in and answer one of the questions that was in the chat. I acknowledge that Olman asked earlier at the start of the meeting if people could ask the questions orally. But while you're thinking of your oral question, Ananda asked, did we use statistical models that considered pedigree and relatedness matrices? And I didn't, I did present the relationship matrix as a heat map in one of my slides, and that's how we discovered that there were 7 small full subfamilies amongst the clones that we thought were unrelated. In my other work not with teak, but with other species, once we have hundreds of individuals, and especially ones that we think are the parents based on the pedigree and the ones we think are the progeny, and then we can start assembling doing the pedigree correction and assembling the genomic relationship matrix in a more complete way. But in this sense, we only had 26 clones that we thought were unrelated, so it's not really enough material. I think it'd be a question for Olman and Jose Pablo whether they want to go that way in the future with their GENFORES population. And now I'll open it back up for oral questions, which is, I think, what Olman was hoping for.
Lupita, if you want to organize, I'm not sure how to do this now, the questions from the people, from the audience.
Yes, Olman. If any has any question, please raise your hands to enable the microphone.
Okay, so guys, if you have, like Lupita said, any of you, if you want to know something more about this, more information or questions, raise your hands, please.
No, Olman. No question.
No, I'm sorry. Let me wait.
Okay, your leg.
I have a question, Olman. This is Emmanuel.
Oh, Emma.
I work in Olman and Jiménez's team, and I would like to ask Andrew, how many extra genotypes should be sequenced using the I may guess that it would be the same strategy that Jose Pablo used for sequencing to get the SNPs. So how many genotypes should be sequenced, and how many environments should be included to have a more robust analysis of the association of SNPs markers? Well, in my case, probably Dawa and I will be the guys working in the lab, trying to convert these SNPs into genetic markers using we have to find the suitable platform to genotype these SNPs. But and actually at the beginning the idea was to convert the genotypes that you already found into genetic markers. But Olman said, okay, let's wait for a while, and because we are going to sequence more genotypes. So that's my question. How many genotypes should be included in the next step? Thank you.
Yeah, sure. And so, obviously I'm not the one paying for it, so I'm going to say 5,000, sure, but to be realistic and pragmatic, I want to probably see, it's a round number, say 100 clones before I start saying the result is preliminary and promising, but the more genotypes that can be assayed, then the more confidence we have in the entire in the entire thing. So from a GENFORES perspective, You know, if you can, I wouldn't say 100 should be the target. I'd say 100 is the minimum, and you should target. Well, I think Olman put a table up with 364 or something clones in the cooperative. That should be perhaps the target to begin with. Because the more you get, and the broader range of true clone values you get, then the more power we have on those correlations and regressions. In terms of the marker panel, I'll just say briefly, I think that, you know, nearly 11,000 SNPs is fabulous. I think that's a good size marker panel for this kind of exercise. And as more clones get genotyped, we'll find, as Jose Pablo suggested, that number will actually increase because he's got more markers in the library. It's just that a lot of them dropped out with minor allele frequencies, and they'll get picked up. And then on the number of environments, it's really a question for understanding the G by E that is affecting that's genotype by environment interaction. Which is affecting the growth of teak in your target population of environments. And so with my very limited understanding of Costa Rica, I'd say, well, we definitely need 1 or 2 sites from Guanacaste, which is drier, perhaps a second site from central Costa Rica, which is wetter, one from southern Costa Rica. So basically representing the different zones that you've got. And then what you're probably going to find is that a clone that had a high clone value for volume at this one site, it might fall out because it was specifically good at this one site and is not good across all sites. So what we end up with right now is a preliminary clone selection for performance at this one site and at sites that are similar to it. If we want clone selection for clones that are high performers across the full diversity of sites, then we need to put that information into the model in the form of a clone value across the sites.
Okay. Okay, thank you. Yeah, one of the last things that you said is about the use to clone the clones that have been selected to be more, maybe to be more precise in the selection of SNPs. As years ago, I had an experience with a selection of SNPs for market-assisted selection in oil palm. And I remember that we selected samples for sequencing, but using a panel of samples that had the largest possible diversity in the germplasms. Should be the same done for teak, because Olman and Jorleni are very advanced in the breeding program, so probably they now have the best of teak, and they now don't have the I don't know if they have already or not the, let's say, the worst ones, and that can be included in the next step to see if some spurious associations can be found.
Yeah,
could be found.
I agree 100%, and especially with marker discovery, you know, including some samples that we know are poor performers is a very good idea.
Yeah, excellent. Yeah, exactly. Very exactly right, Emmanuel. Emmanuel is part of our team in the genomics, in the molecular genomics work, and he works for another national institution in biotechnology in Costa Rica, founded by the public universities, and he has been coming back. Actually, Emmanuel was part of our GENFORES at the beginning, but he moved to the private companies. Now we are getting him back, and so I'm very happy. And he is exactly right in that analysis of not only looking at the the winners, let's say in an easy way. You have a ranking, who are the winners, but it's going to be also very important to know who are in the lower positions in the ranking and look at their DNA. I mean, you can get into all this business of prediction analysis, sometimes you can know who are the bad guys. And actually, in philosophy, if you go into real sciences, when you raise a hypothesis, the only thing you do in real science is to answer to get the, let's say in an easy way, right? To get the bad ones. You don't know, and then you continue with a new hypothesis. This is the whole philosophy of sciences. So sometimes it's probably easier to know who are the bad guys. Like, well, we have that now in the politics very easily, you know all of that. So sometimes very easy to know who are really the bad guys, and not that easy, and could be very difficult to know which are going to be the real winners. So this trade-off is very good, Emma. Your question is very good.
Thank you, Andrew and Olman, for the answers.
We have a couple questions in the chat. Go ahead and read the first one. It says, from Enrique Garcia, is it possible to find a clone with traits related with shorter rotation period? On that matter, I'll say that what we're doing is try to shorten the breeding cycle by enabling early selection. However, I don't think that growth rate was one of the traits that were analyzed on this, and Andrew will correct me if it is. You could potentially find any association with growth rate and select traits that grow faster. But if you're looking at the breeding cycle as it is, the best way would be early selection to cut down on those times. If we can do genomic prediction and select in the nursery rather than 7-8 years after in the field trials, then we're shortening that breeding cycle significantly.
Yep.
Yeah.
So sorry, I hope you're finished there, Jose Pablo.
Yeah,
just
go ahead.
To add to that, analyzing for volume and selecting clones with a faster growth rate by increased diameter, increased volume, increased height, then you got two choices. You can either leave rotation length where it is and have larger trees at rotation, or you can bring the rotation shorter, and with just a consideration of whether the wood properties are adequate at the shorter rotation. But that decision is purely economic and market driven.
And the other one that we have here is, once you find the Evelyn Bush, the Chacon asks, once you find the genetic material desired, what method of production or technique are you using to multiply the material? And here I'll defer to Olman and maybe Jordi, if it is around, she's around.
Talk about the vegetative.
Well, now, yeah, thank you. Now with the clonal development techniques we have, it is extremely easy to do it through clonal development. Clonal development as in general. It's quite easy for teak. It is a very robust plant, easy to propagate, and we have very good propagation parameters. This is really, and it's amazing. It's even less expensive than traditional seeds. This is because of the technology, and we have been very active in the last 3 years, precisely in that area. How to reduce costs for producing mass scale clonal propagation of teak and some other species, of course. And so our numbers are going down and down every time. We are getting into very low numbers for producing mass production of selected material. And that is what we call clonal forestry. And all those work together with the breeding programs, of course. Actually, clonal forestry, your question is correct, is the end up of a good breeding program. You translate all the efforts, all what we have been talking today, genomics, breeding, but at the end, what you will have is very good genetic material into little cuttings, little plants, the plants that go to the field. That's basically the end up of a very good, very well organized breeding genomics programs. I'm through clones, because if you don't clone it, then you lose a lot of information. Just remember that basic principle. If you okay, you already know who is going to be a superior tree. Let's say that you predict very nicely this tree or this plant is going to be the best, the winner. Yeah, good. If you don't take the same plant and propagate it thousands of times, then you're losing that information immediately. If you take the seed, for example, from a +3, the seed will be only 0.5% of that seed will carry the genetic information from that super tree that you are looking at. And that is a mother. And the father, you don't know where it comes from. So we always have to go, the best way to do all this, to take advantage of this information, is to go through clonal propagation, because you are going to replicate that tree that you already know that is superior. See?
Gustavo.
Hi, thank you. Thank you, you all for your presentations. They were very interesting. I'm Gustavo Hernandez from National University, from INICE4. I got one question, basically is, well, it maybe it's a ask for a recommendation. You said that from the clones that you studied, they were very related between each other. And my question is, if you could give a recommendation to the companies that are part of GENFORES for them to continue improving the genetic improvement programs, what would that be regarding these high related clones that they have in the programs? Thank you.
Andrew, let me just say one line, and you and Pablo will do much better than I. I'm from a very old breeder generations. So with this information, Gustavo, as a breeder, as an old breeder, I would say, well, beautiful information. I would take care of how to conform these groups. When you go through the mating job, when you are in advanced breeding generation, you want to combine genotypes unrelated, because you want to promote a higher, this is the classical breeding stuff that you get when you are learning breeding. But now with genomics, then I will let Andrew and Jose Pablo talk to you about genomic selection. This is completely, to me, a new world. The new genetics is coming now. When you have such a beautiful information, and now you are not going to be using the classical 25% half SIPs, full SIPs, quantitative classical genetics. That is completely going to be lost. They are now going to talk about the relatedness. You have a 40%, 45%, 55, such more beautiful, detailed information about the kinship, about the relationship between in your breeding population. So now it's a completely new world. I'm happy I'm going almost to retire in a few years, because this is completely, to me, is too much to me for my mind and as an old breeder. So these two guys will explain that what is going to be genomic selection, and how to use it better, all these mating, pair mating, all this, how to mate all these materials in a breeding population.
So firstly, from a practical perspective, a clone with a high clone value still has that clone value, whether it's the full SIB of another clone with a high clone value. It doesn't diminish the value of the clone to know that it's related to something else. It might be of practical importance if a company thinks it's deploying 10 different clones for risk management, and it's got a policy of only deploying 10 clones, and then they find 3 of them are related. Well, then that is of importance, because it has an impact on risk with the narrower genetic diversity of clones. So the biggest impacts of knowing these relationships come back to what Olman was hinting at, which is that when we come to breeding, and it could be open pollinated breeding, breeding with half SIB families from OP, or it could be control pollinated. But in either case, it's really important to know the relationships amongst the parents. So it has profound effects on the genetic values that you attribute to the different progeny once you measure them. I'll just leave it there.
I'll just add, as Andrew said, again, yeah, being related doesn't diminish the value of any given clone. If it is a good clone, it will continue to be a good clone. I think that what we can say from this is that with genomic information, we jump from this traditional view, right? Those expected values given a pedigree to what we call a realized relation. So you know exactly how similar two clones are, even if they're related. Because I mean, we assume that you share 50% with your siblings, but which 50%, right? What part of that? Genomic selection gives us a better understanding of how much are those related, how similar are, and that should inform your crossing definitely. I mean, you want to breed in new versions or new diversity into it. So you have to take into consideration the current or the realized relationship between the material that you have. But that's it.
Just one last important information for Gustavo and for the audience. This is not going to happen. It's not going to be really a risk result in the very short term. In the next generation, in the next breeding generation, the F2, let's say, to me, is not going to be a big deal. After that, yes. And not only after that. And if you want to focus on specific traits, if you really want to move faster, let's say, for example, in wood properties or something like that, or a disease resistance, there, I would say, yes, the genomic information is going to be highly valuable now. But in general, I mean, relatedness is not going to be a big deal one more generation. But after, in the long term, yes, it's going to be extremely important. Except, again, if you want to focus on now, and go fast with some specific goals. There is going to be now, not in the future. Now it's going to be very useful to know all this relatedness.
Raymond.
Yes.
Raymond, sorry, no, just to tell everybody, and I don't know if you know who is Raymond Keogh, maybe the father of teak in the world. Raymond, really welcome. I'm very glad to have you here.
Thank you very much, Olman. You can hear me well?
Very, very well. Go ahead.
Good. Well, you talk about age, Olman. I think I'm a previous generation to yourself, so don't give up quite yet.
No, thank you, Raymond. Yeah, I know you. We are really pleased to have you. I'm very happy to see you so well.
Thank you very much indeed. Just a quick point. I was very interested to hear somebody talk about, you're obviously concentrating on the better material and for commercial purposes, etc. Absolutely. Don't disagree with that. Now, the other point that was made is, well, what about the bad stuff? And I have a point to make on this one that might be of relevance to you. And that is, in the old days, I did quite a lot of work on tracing the pedigree of teak in Central and South America. And one of the things that came up was quite fascinating, was the low level of points from which distribution of teak into Latin America came about. And one was that famous Tenacerum Trinidad strain. And we know that kind of came into, I came across it in Honduras and El Salvador, etc. And another one is the Summit Garden, Paradenia Summit Garden, and Summit Garden distributed this all over the place. Now, I know there are still trees outside the Summit Gardens that are I think, the original material that was brought in from Sri Lanka, Paradenia Gardens in the 1920s. And what do you think about what is the value of looking at these trees? In other words, to get the question that you're saying, well, where does the material come from? What is the value of looking at those trees and getting a genetic fingerprinting of those and the Tenacerum Trinidad variety, and just sort of so that when you're broadening the studies, you'll be able to say, got this. Now we know exactly where these things are coming from. Just an idea from the past.
Well, I think all of us can answer you, Raymond. Well, I will always remember your invitation to do all this trace back from the origins of the teak in the whole continent, American continent, and also in the African continent. Now with this genomics, we can do a lot better that type of analysis. As we all know, there are some good jobs, some studies done about the African origins, the origins of the teak in Africa. We have not done such a nice work for the Latin American teak. And as you mentioned, well, for all of you, Raymond has been the guy tracing back all this information, what he was exactly mentioning, the origins from the British probably were the ones who brought the material through Trinidad and Tobago, but there were some other sources as well. And Raymond was also in relating I remember some types of leaves very clearly directed to some provenances. And that is a beautiful work that Raymond did for many years. Now, with genomics, we can do a lot better than that, and Raymond, exactly the same. Now, what is the importance of this to me as a breeder? In a long way, we have to think in many other situations that are happening, like climate change, for example. Then if you are only focusing in the short term, you're lost, especially in breeding. You can't be focused only on what is the winners in growth and yield and so on. No. We're going to have much more difficult situations like climate change, like pests and diseases, like, and as a good friend of mine, Volker from Pan American Woods, he taught me one beautiful sentence. This is not a business of good-looking trees. This is a business, teak is a business of very highly valuable wood. So the wood, you may have a beautiful tree, but the wood could be not that special. So this is another important issue that Andrew was pointing very well. Wood properties is a key factor in teak business. So I'm just trying to answer Raymond. Tracing back is really important. We still have to do that job. We can now do it better with the genomics. Of course, it is gonna be directly. And secondly, guys, remember, climate change is coming. Not is coming, it's already there. Pests and disease, we already have a permanent working line in disease resistance. We didn't have that 20 years ago. Now it is a reality. And in that area, you need to keep larger populations, original population, and there is where you go back to try to get some answers and some help. And the Brazilians helped learn and teach us. They show that in eucalyptus, and that is another question related to Raymond's question. In eucalyptus, they found that crossing completely different species of eucalyptus is producing a lot better than working only inside one specific eucalyptus type. So, well, Andrew is an Australian, and Andrew knows more than I, especially this area of eucalyptus. But that's another something to learn. So I've been also interested in the Brazilians asked me once, are there any other teak related species like teak genus and Tectona something? I say, well, I think there is one I've never heard exactly. Maybe the Asian guys who are the owners of the natural teak population may know about that. And so I will put that also in the same type of question that Raymond is raising. So let's only look at the short term of the time. The long term is going to be really very important as well.
Agree.
Thanks for your point, Raymond. I think it's very well made. And it's certainly the kind of thing we're doing in other species, where we have verified individuals of different provenances or races, and we're genotyping those on the same marker platforms as our commercial material to trace back those relationships and to learn the kinds of things Olman was talking about. It's an important part of tree improvement strategy to know where the different centers of variation have come from across the natural population, and variation in the key traits that we're trying to improve, as well as the history, which is an important part of the legacy of the material we're working with. The only thing I'd really add is that we can find published reports from genomic studies of where the origins of different material have been. But in order for it to be commercially applicable information in GENFORES, we need to genotype those reference individuals with the same marker panel that GENFORES is using. So that's the action item to go on the list. Yeah.
Right.
I'll only add to.
There are still, sorry, just a last point, there are still trees, for example, as I mentioned, Summit Gardens, but in Patadenia Gardens, there are also a couple of trees there, which could be the pedigree line of those in Panama and elsewhere.
Yeah, well, we should definitely look into include as many of those trees that we can, just to get an idea, as you say, trace back that pedigree of all the individuals that we have in our breeding population. And we also need to remember that the base of any breeding program is a conservation program, right?
Yeah.
As someone mentioned before, those not so nice or not so pretty trees right now might not have a value that we see, but as climate change and future conditions change, those trees might provide valuable traits that we need to introgress or bring back into our breeding population. So the more trees that we can map, the more trees that we can genotype, that we can keep in our germplasm banks, the better, because we have a broader diversity, a genetic diversity to choose from, right? And that's something that we have to worry about, because every breeding program, be it teak or be crops or whatever, they're suffering from what we call genetic erosion. Like after several cycles of selection, you're reducing the diversity, right? So we need to be aware of that and have material to bring it back to introduce more diversity into it. Or we end up with some crops that they have to do it by mutagenesis or create the diversity because they already ran out. And there's no gain to do if you don't have any diversity to choose from.
I'm sorry. Is there any other question? If somebody wants to raise a question, you can write it in your language if you want, in Spanish or Portuguese or whatever, and then we can translate it into English if you want. If you can put it in the chat.
I have one question.
Go ahead.
Okay, my name is Ananda Aguiar. I work in Brapa Floresta in Brazil. I would like to know if you intend to work with phenomic and environmentomic data. Because everybody talking in Brazil right now, and United States, about getting in the breed program a lot of crops. And I think that maybe Phenomica has maybe the same result that genome.
What do you think, Andrew?
Just to clarify, Ananda, I think you're asking about the enviromics. In other words, capturing the environmental signature of the different sites with a large number of environmental variables. Is that what you're talking about?
Exactly. Sorry about my English.
No,
exactly. Is the data of you can put together a set of the genomic and trait and climate variables.
Yeah, perfect. No, that came through perfectly. I just wanted to check. Look, in a separate study that I'm involved with and will get published later this year, not with teak, but with eucalyptus, it's been extremely powerful, extremely powerful approach, and I'm a huge advocate of it. I'm glad that you brought it up. I can't really speak to the potential for implementing it in GENFORES because really the approach needs high quality environmental data. The climate data that we can get from public sources is now very good. But the soils data, what I found from my study in Australia is the soils data from the public databases was not good enough. And so it needed direct inspection, laboratory analysis, and directly inspected pits in order to work. So I'll hand it over to the GENFORES people and see if that's a potential for this this breeding population.
Okay, thank you. Because here in Brazil, the people can accumulate a lot of data set, not just a progeny test or clone test, but also for the plant, commercial plant, and try to put together, for example, right now I try to do it for Pineteda and also Araucaria, and put together and try to correlate with a different climate. But I know that in Brazil don't have the good data set about this soy, for example, Kinko, everything. Do you know about it? It's very hard to get it.
Sure.
But I believe that you can use another information for example, for the companies, forest company here in Brazil, I can get more information about a lot of different trait and genetical control.
Ananda, if you're actively working on this, we can connect separately after the workshop if you're interested.
Sorry, can you repeat, please? I don't have a good connection.
I was just saying I will look you up and send you an email about this directly after the meeting.
Thank you.
Thank you, Ananda. Obrigado.
De nada.
Any other question? No question, Olman.
No questions? No more questions?
No more question.
So, guys, and all the friends, thank you. We are really thankful for your time, for your listeners, and well, to help us a little bit to share all this interesting information. This is the idea, to share with you where we are, where are we going to, and we will be like always very open to any of you. You can write us, you can send us messages for more information, of course, but it's going to be the idea. I'm not quite sure if they have that our mails or something. I don't know. We're not sure if people want to write us some mails and how to do it. I don't know how to. We may try to point out somehow our mails. Maybe, I don't know, because that could be, we should do that. I don't know. I don't see that, for example. Now, if somebody want to send us a message, how to do it, I don't know. This is so
we have the emails.
uh-huh.
Yes.
So people can get our mails from the general information. Yeah. Okay. Then it's great. So guys, I don't know, Andrew and Jose Pablo, if you want to say something else. I'm finished or and really happy with having people, especially Raymond Kyo. Yeah, I really am really happy to see you so well. You are still remember all those details, man. You are. I would like to be like you. In the yes, you are in the other breathing generation. Yeah.
Thank you, Olman.
Thank you.
Yeah, no, I'll just say, yeah, it's great to see Raymond here and many other friends as well. Thank you especially to all of you for hanging on so long through the discussion and question session.
Thank you.
Thanks everyone for your time. Appreciate it.
Thank you, Olman, Jose Pablo, Andrew, for sharing the advanced results of teak genomics. Thank you, everyone, for connecting to this webinar. Yes, we will be sharing the recording and the
Okay.